Okay, in this video we're going to talk about bone remodeling. So about 5 to 7% of your bone's mass is recycled each week. In fact, spongy bones replaced every 3 to 4 years, and compact bones replaced every 10 years. So if you think about yourself 10 years ago, you had different compact bone then, and your spongy bones have been replaced several times since then as well. Now, bone remodeling consists of both resorption and deposit. Remember, bone deposit is mediated by osteoblasts, and bone resorption is mediated by osteoclasts. So we're talking about the building up and the breaking down of bone. Now, these occur at the surfaces of periosteum and endosteum, and you find that these osteoclasts and blasts are located nearby in what we call remodeling units, which are basically packets of blasts and clasts that are nearby, and they coordinate the remodeling process. So bone deposit is when new bony matrix is deposited by osteoblasts. We find that osteoblasts create new osteoid, and we see that there's a band of unmineralized bony matrix near these osteoblasts. What we find then too is that these osteoid seam gives rise to a calcification front where this is actually mineralized osteoid that had been created or laid down, at some time prior, but is now calcified because of the calcium binding proteins and collagen fibers that trap those calcium minerals. Now, the trigger for bone deposit is confirmed and can include things like mechanical signals, increased concentrations of calcium and phosphate, matrix proteins that bind to calcium, as well as appropriate amounts of alkaline phosphatase for mineralization. Now, resorption is the breakdown of bone, and this is a function mediated by osteoclasts. We find that osteoclasts actually dig depressions or grooves as they break down the matrix, and they break down the bony matrix by secreting lysosomal enzymes and protons onto the matrix that actually digest that material. So acidity converts the calcium salts into soluble forms that can dissolve and then float away from bone and dissolve throughout the fluid of your body, including the extracellular fluid and blood plasma. So osteoclasts also phagocytose demineralized matrix and dead osteocytes. These digested products are transcytosed across the cell and released into the interstitial fluid, then into blood. And once resorption is complete, osteoclasts undergo apoptosis or programmed cell death. So these osteoclasts don't live very long. However, their activity is under hormonal control. So we find that osteoclast activation involves parathyroid hormone as well as immune T cell proteins that can stimulate osteoclasts to become more active. So we find that remodeling occurs continuously, but is regulated by genetic factors as well as hormonal factors. So the hormonal controls of remodeling include negative feedback loops that regulate blood calcium levels. So we find that if blood calcium levels are too high, then that's going to stimulate bone deposit, where calcium is stored within bone. Or if blood calcium levels are too low, this is going to stimulate bone resorption. That way you can release some of the calcium from bone into the bloodstream to get your blood calcium levels back up. Now, calcium functions in many important processes, which is why it's one of those things that needs to be tightly homeostatically regulated. And we find that calcium is significant for nerve transmission, muscle contraction, coagulation of blood, and nerve secretions, as well as glandular secretions and cell division. So it's extremely important in all these essential roles. We find that most of the calcium of our body, like 99% of it, is actually found in bone. The rest of it is in solutions of our body or in cells. And intestinal absorption of calcium requires vitamin D. So vitamin D is a hormone that stimulates the intestinal cells to be able to absorb calcium from the food you eat. Now, the second thing that also controls remodeling is actually mechanical stress. So we'll come back to mechanical stress in a little bit, but the other hormonal controls of remodeling include parathyroid hormone and calcitonin. Parathyroid hormone is produced by the parathyroid glands just behind your thyroid gland on your neck, and parathyroid hormone, or PTH, stimulates osteoclasts to resorb bone or break it down. Therefore, calcium is released in blood, and it raises your blood calcium levels up. Once your blood calcium levels are in a normal limit, parathyroid hormone secretion stops, which is why it's a negative feedback loop. Calcitonin is the opposite. In fact, calcitonin is actually released by your thyroid gland, also in your neck, but these are released by the parafollicular cells, and these are released in response to high levels of calcium. The effects are typically negligible, but at high pharmacological doses, it can lower blood calcium levels temporarily. So calcitonin is something that's synthesized and secreted by our bodies, and it's been shown to lower blood calcium levels by stimulating bone deposit. So you're removing calcium from blood and basically storing it in bone. So this is showing this nice little negative feedback loop of blood calcium homeostasis. We find that we want to maintain about 9 to 11 milligrams per 100 mils in our body, and if you get outside of this normal limit, like if your blood calcium levels get too low, your parathyroid glands can release parathyroid hormone, which travels in your bloodstream and stimulates osteoclasts to become more active. Once these osteoclasts are more active, they can resorb more bone, break down the matrix, release that calcium in your blood, and then get your blood calcium levels back up. Once calcium homeostasis is returned to like normal physiological limits, then we find that parathyroid hormone release stops. So you stop stimulating osteoclasts. So other hormonal controls of bone remodeling include things like leptin and serotonin. Leptin is actually a hormone released by adipose tissue, but it plays a role in bone density regulation by inhibiting osteoblasts. So you find that if someone's leptin levels are too high for an extended period of time, then you're going to inhibit bone deposit, which can lead to things like osteoporosis. Now, serotonin is a neurotransmitter that regulates mood and sleep, but it's also something that can interfere with osteoblast activity. So most of the body's serotonin is made by your gut neurons, and it's actually involved with coordinating digestive activities, but it's secreted into blood after a meal. So a meal after a meal, serotonin levels rise in the blood, and it may inhibit bone turnover after a meal, so bone calcium is locked when new calcium is flooding in the bloodstream. So if your serotonin levels are too high, it actually prevents calcium from being essentially released from bone. Now, other factors that can affect remodeling aren't just hormones, but also responses to mechanical stress. In fact, bones will reflect the stresses that they encounter. So that bones that are stressed when weight bears on them and muscles pull on them, and essentially they're going to regrow and remodel in a way that reflects the type of stress that's been introduced to that bone. In fact, we call this Wolff's Law. So Wolff's Law tells us that bones grow and remodel in response to the demands placed on them. Stress is usually off-center, so bones tend to bend. Bending compresses to one side and stretches on the other side. So the diaphysis will be thicker on the side where the bending stresses are the greatest, which makes sense because you want the bone to be able to resist that stress. And the bone can be hollow because more compression and tension cancel each other out in the center of that bone. So what we find is that the medullary cavity stresses and tensions cancel out, which explains why there is no spongy bone in the medullary cavity. So this shows an example of stress that's placed on your femur. So because of your body's weight here, you find that the lines of stress in your femur are kind of at an angle. And you see there's a compression force on the medial side and a tension force on the lateral side. But these stresses and tensions cancel out in this midpoint here, which explains why there's no spongy bone there. You have a medullary cavity because there's nothing to resist here. However, the compact bone in these regions is much thicker in order to resist these tension forces and compression forces that are placed on the bone. In fact, the more body weight and the more stress you place on this bone, the thicker those bones are going to get in order to resist those stresses. So Wolff's Law explains the hardness of bone. So you find that with handedness, the hand that you use the most, whether you're right-handed or left-handed, results in thicker and stronger bones that correspond with that hand. So right-handed individuals will have thicker bones on the right side of their body because they're more likely to use their right hand for a lot of tasks. Now, curved bones are thickest where they're most likely to buckle. Trabeculae, or basically the spongy bone in these bones, can form trusses along lines of stress. And large bony projections occur where heavy, active muscles attach. In fact, when muscles get larger, bones get larger where those muscles pull. So weightlifters have enormous thickenings at muscle attachment sites. And this is useful for muscles because they have more bone to attach to, and the bones can actually resist those stresses placed on them. Now, the bones of a fetus and bedridden are usually featureless because they lack stress on the bones, and the bones didn't grow or remodel in response to that particular stimulus, so they're practically featureless. Now, mechanical stresses cause remodeling by producing electrical signals when the bone's deformed. We'll go into more detail what these electrical signals are when we get to the nervous system, but we know that the movement of fluids with lots of ions in it creates a separation of charge, and these oppositely charged regions are electrical signals that osteocytes can use to help stimulate remodeling. So you find that fluid flows within canaliculi, and this can stimulate remodeling in a way that allows for the bones to grow and remodel and to basically resist the stresses placed on them. Hormonal controls determine whether and when remodeling occurs, and this can also affect blood calcium levels, but mechanical stresses determine where it occurs, specifically where in the bone will that bone get thinner or thicker, and it's based on the mechanical stresses or demands placed on it.